Okay. I promise I will get them back to you as quickly as I get them from the TAs, and that's what I'm waiting on at the moment. They have an exam, but as I told them, I still expected them to have it done before now, so that's life. All right, so we're going to move forward with fatty acid oxidation. We've got fatty acid oxidation, we've got synthesis, and we've got ketone bodies and also prostaglandins to talk about. So we've got quite a few things to talk about between now and Wednesday, and so I'm fairly optimistic. I think we're actually in pretty good shape with stuff here, but we have a fair amount of material to cover. I know you guys know I go fast, and I am going kind of fast through things, so I apologize for that, but we are partly, because of our off day, a little behind. Okay, well, last time I talked about the fact that we have the problem with fat, and that fat is water insoluble, and you've already seen the issues that the body has with respect to moving that insoluble material through the bloodstream. And we also recognize that that insolubility requires a good storage mechanism. And so, as I mentioned last time, there are specialized cells called adipocytes that are involved in storing that fat. What you see on the screen is a depiction of the breakdown process for a fat, and that is involving using a lipase. And a lipase, right here, cleaves each fatty acid off of the glycerol backbone, leaving glycerol plus 3 fatty acids. I told you that lipases can be extracellular, and lipases can be intracellular, and what I'm going to focus on today are all things that are happening inside of the cell. So everything I'll be talking about today will be happening inside of the cell. When we look at the regulation of fat breakdown, we see, not surprisingly, that fat breakdown is partly a function of a system like you've seen before. It's regulated through a 7-TM. That 7-TM is the same 7-TM that you saw that was involved in the regulation, the breakdown of glycogen, and also in the glucose and gluconeogenesis, glycolysis and gluconeogenesis pathways. This is the beta-adrenergic receptor. We have binding of a hormone to the receptor, which activates a G protein, which activates adenylate cyclase, which makes cyclic AMP, activates protein kinase, and protein kinase comes in and acts in a couple of places, and I don't think we need to know all the detail that's in here. This is a little bit more than we really need to know. But what I would say, too, that I think that you should know is that the phosphorylation of a target lipase activates that lipase, and thus activates the breakdown of fat. So the breakdown of fat is occurring here, excuse me, inside of an adipocyte. And that adipocyte could release those fatty acids and glycerol out into the bloodstream. And again, the fatty acids are going to be carried by the serum albumin. I can't pull the word out myself. Carried by the serum albumin in the blood. And that serum albumin will then donate those fatty acids to target cells so that the cells have what they need. Okay? Well, what we saw in the case of glycogen breakdown was that phosphorylation of the enzymes involved in glycogen breakdown tended to activate them. And so phosphorylation activated breakdown, and phosphorylation here is also activating breakdown. Okay? So, not surprisingly, breakdown is favoring the phosphorylation is favoring the breakdown of larger substances to smaller substances, and of course that's what's happened, that's what's necessary for energy production. So again, cells are needing energy. This turns out to be the only regulation of fatty acid breakdown. Okay? Fatty acids themselves, there's no regulation of the scheme that I'm getting ready to show you. So the fatty acid breakdown is controlled at the level of the breakdown of fat. Okay? It's controlled at the level of the breakdown of fat. Now, we will see some regulation in the synthesis of fatty acids. There's one primary regulatory step. But for our purposes, the breakdown of fatty acids is only regulated at the level of breakdown of fat. Glycerol itself is another component that is produced by fat and by the breakdown of fat. And glycerol turns out to be the only component, with a minor exception that we'll see later, but it's the only component that can be used to make glucose out of fat. Okay? Glycerol is a 3-carbon compound. That 3-carbon compound can be converted into in this case glycerol phosphate, and ultimately into dihydroxyacetone phosphate. Well, of course, once we get to dihydroxyacetone phosphate, we can make glucose through gluconeogenesis, and so glycerol can ultimately, if we have two of them, glycerol can ultimately be converted into glucose, and that's the only component, the only significant component of fat that can be. The fatty acids are largely broken down to acetyl-CoAs, and acetyl-CoA, I hope you learned from earlier, is not a source of material for making glucose, at least in animal cells, because we don't have the glyoxylate cycle. Plants can use that, but we can't do that. Okay, don't think we need to go through that. All right, so let's turn our attention now to the breakdown of individual fatty acids. I gave you a schematic showing you that the other day, and I want to just briefly show you that again to remind you. So when we look at fatty acid oxidation, this process is shown here on the left, and it's a very nice scheme. I like this overall view of this picture of this process very well. And this process has a name. It's called beta oxidation. Okay? Beta oxidation. And the reason is because if we number the carbons as alpha, beta, then all the action happens, as I said, between carbons 2 and 3. It's the beta carbon where the process is happening, as we shall see. Okay? So to remind you, there are several steps in the process. The first step being an oxidation step, and that oxidation step is known as a dehydrogenation. Okay? A dehydrogenation. You've seen dehydrogenation before, as I said, but this is the similar dehydrogenation to what you saw in the succinate dehydrogenase reaction, pulling hydrogens and associated electrons away from two carbons, leaving behind a double bond. Notice that double bond is in the trans configuration. And again, that's exactly what happened in the synthesis of fumarate in the citric acid cycle. Hydration followed that. That takes a water, splits it into an OH and an H. The H goes on to carbon 2. The OH goes on to carbon number 3. Okay? And again, that's very much like what we see in the citric acid cycle, where we convert fumarate into malate. That hydroxyl group is a target for further oxidation, and that further oxidation yields this compound, and that is equivalent to the oxidation that we see in the citric acid cycle for making oxaloacetate. So again, these things are very, very similar. And then the last step in the process involves what's called thiolytic cleavage, Thiolytic. And that process uses an enzyme that I'll talk more about later. That enzyme is known as thiolase, Thiolase. Now, there's only just a couple of enzymes I'm going to ask you to know the names of. Thiolase is one of them. So thiolase catalyzes this last step where a acetyl-CoA is split off and a fatty acid with two fewer carbons is left. Now, in each case, this says an R, 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 R. These R's are all coenzyme A's. Okay? In the case of oxidation, they're all coenzyme A's. So that's the overview. Let me take you and just say a couple of words about the individual steps to consider in this process. Okay, let's imagine that I am a cell that has taken in a fatty acid from outside. It's been delivered to me by serum albumin, and I've gotten it inside. Okay? Well, that fatty acid, when it gets inside the cell, is just a free fatty acid. And as I've tried to point out to you before, that's a little bit of a problem because this guy can act as a detergent. We don't want it acting as a detergent and denaturing our proteins. So cells basically cover it up with a CoA on the end. And that's what's happening in this process. That takes energy to do so. You'll notice it takes a lot of energy. It takes two phosphates off of there. ATP is going to AMP to yield what's called an Acyl-CoA, ACYL. So the acyl is an acyl group. It just simply means it's a fatty acid. So a fatty acid, CoA, is what's produced by that. Okay? Now, fatty acids with a CoA on them, unfortunately, don't make it across the mitochondrial inner membrane. So the cell puts a CoA on there, and then it takes it to the mitochondrion and goes, uh-oh, a CoA won't get me across the membrane. So the cell has an enzyme known as a carnitine acyltransferase that will take that acetyl-CoA, it'll take off the CoA it just put on there, and it will replace it with a carnitine group. So it's basically making an acylcarnitine. It turns out that acylcarnitine is, in fact, something that can be transported across the mitochondrial inner membrane. There is a translocase that does that. We can see that happening here. We see that translocase is an antiport, and the antiport, when it brings in an acylcarnitine, kicks back out a carnitine. There's a one-to-one relationship, and so we have accounting for all the carnitines that are there. Then, and of course, fatty acid oxidation, as I've said before, occurs in the mitochondrial in the matrix of the mitochondrion. Once the acylcarnitine has gotten in, the carnitine is removed and replaced by a CoA. So it's kind of a dumb process. Fatty acid comes in, acyl group gets attached, acyl group gets removed, replaced by carnitine, carnitine comes in, carnitine removed, replaced by CoA. So in essence, what we've got at this point is an acyl-CoA that is in the matrix of the mitochondrion and ready for oxidation. Okay, so we're ready to oxidize this guy. Jody?
Is this in all cells?
This is in all cells, yeah. Yep. Okay. So here we go. Oxidation. So we've seen the oxidation process. That's exactly the figure that I showed you before. It's now showing you a little bit more detail. And the detail is that when we have an oxidation, of course, we have to have an electron acceptor. And the electron acceptor for this first dehydrogenation is FAD. It becomes FADH2, just again like we saw with the succinate dehydrogenation reaction. So we have a product of an FADH2. I don't care that you know the names of these guys. That's not important. All right? Trans delta 2 enonyl-CoA, L3 hydroxyacyl-CoA. I'm not worried about that. But I think you should know their general structure. You should know that there is a double bond in the trans configuration between carbons 2 and 3. You should know everything is happening between carbons 2 and 3. The water, when it adds, the hydroxyl group goes on to carbon 3, and it creates an L structure. We will see in fatty acid synthesis that there's a D instead. That's one way in which they differ. So we have an L-configured hydroxyl group on this at this point. The oxidation of that hydroxyl group causes NAD to be converted to NADH. So at this point, in this oxidation of this fatty acid, we've made one FAD and we've made one NADH. We are then ready for thiolytic cleavage, and thiolytic cleavage happens right between carbons 2 and 3, and we have the products that we talked about before. Now, there's only one enzyme besides thiolase on here I want you to know, and it's an important one. It's the very first one. It's called Acylcarnitine dehydrogenase. Not a complicated name. Acylcarnitine dehydrogenase. So it's working on an acyl-CoA. It's removing hydrogens and electrons. It's making FADH2. Why do I want you to know that name? Well, it turns out to be an important enzyme from a health perspective. Okay? Our bodies have three different forms of this enzyme. One that works on long fatty acids, long meaning longer than probably about 18 or so carbons. One that works on medium-length fatty acids, and it'll typically work on fatty acids anywhere from about 8 to 18, 8 to 16, thereabouts. Okay? And then there's the ones that work on short fatty acids, and it'll work on fatty acids generally shorter than about 8 carbons in length. All right? 3 different acyl-CoA dehydrogenases. Okay? And as we'll see, the longer one is found in a different location. It actually is not in the mitochondrion, but we'll talk about that in just a bit. It's the medium one that's of interest from a human health perspective because it's the medium one that is frequently found to be deficient in children who die of sudden infant death syndrome. So it appears, at least in some cases, that sudden infant death syndrome is linked to an inability to break fatty acids down through that middle component. They're getting plenty to eat, and so on and so forth, but they're not breaking fatty acids down through that middle range. As a consequence, probably are starved for energy at that point. Okay. Let's see. That process will continue and continue and continue until finally we break everything down. Now, fatty acids, when we look at them, we discover that they mostly have even numbers of carbons. Mostly they have even numbers of carbons. We're going to see some other considerations, but mostly they have even numbers of carbons. So if we start with 18, it goes 16, 14, 12, 10, 8, 6, 4, and when it gets to that 4, it splits it in half, and you've got your final 2. Okay? If we have an odd number of carbons, we have other considerations, and I'll show you that. Okay? There's a bunch of enzyme names that we're not going to worry about. And as I said, the acetyl-CoA dehydrogenase and the what I call thiolase, they call it beta-ketothiolase. You can call it thiolase, and that'll be fine by me, are the 2 enzymes whose names I think you should know. Okay, now, oxidation of fatty acids is important, and it's an important source of energy, but of course, what you've seen so far that I've shown you has been the oxidation of fatty acids that have saturated bonds. Okay? I haven't shown you any unsaturated bonds, so we have to have some considerations for how we oxidize fatty acids with unsaturation within them. An example of that might be the one that you see on the screen known as palmitoyl-CoA. Palmitoyl-CoA has 16 carbons, and it has a single double bond, as you can see here. Well, if you think back to what I showed you before, you said, well, there was a double bond that was in that thing that you showed us. Can't it just go from there? And the answer is no, it can't. Okay? A couple reasons. One is a double bond may not be positioned in the right position, number 2 and 3 that I talked about. That's one consideration. And the other consideration is the double bond here, as it exists in most fatty acids in our body, is in the cis configuration. And the one that you saw before was in the trans. So there are some considerations in then how a natural cis-containing bond of an unsaturated fatty acid must be dealt with in fatty acid oxidation. That's what we're getting ready to see right here. Okay? All right. Here's what happens. All right, so we go through several cycles, each cycle knocking off 2 carbons at a time until we get to this thing that looks like this. All right? So here's carbon number 1, carbon number 2, carbon number 3, carbon number 4. First of all, the double bond, we notice, is not between carbons number 2 and 3. And second of all, it's in the cis configuration. Well, this arrangement turns out to have a very, very simple solution for the cell to consider. It simply moves the double bond from position 3 and 4 in the cis to positions 2 and 3 in the trans. Bang! One enzyme. And this guy now can go through the rest of beta oxidation without problem, because the next thing that's going to happen to this guy is going to be what? Addition of water, right? So it'll have water across this, and we'll continue along the process. It'll be oxidized just fine, and everybody's fine and dandy. Okay? Well, things aren't always that simple. What if we have multiple double bonds, and they have other arrangements? And so that's what we have to talk about in our next consideration. I think you should know the name of this enzyme. It's known as enoyl-CoA isomerase. I don't care if you know the cis part, but enoyl-CoA isomerase. That's the enzyme that converts a 3,4 cis to a 2,3 trans. That's going to be an important consideration in fatty acid oxidation overall for unsaturated fatty acids. We'll see how that comes up in other places. Okay, what if we have something that looks like this guy? Okay, and with this guy, we have 2 double bonds. And they're not positioned exactly the way that we need to have them positioned. Okay? So how do we deal with this? Well, let's take this into consideration. We, first of all, have a linoleal, can't even say that, CoA. All right? And we go through several rounds of oxidation until we get to this process. Okay? We're sitting right here. We've got 1, 2, 3, 4. And we see, what do we have? We have a 3,4 with a cis, and we want to make it into a 2,3 with a trans. There's our friend from the last cycle, enonyl-CoA isomerase. It does exactly that. The next step that will happen to this is we will add water across this double bond. Okay? And when we add water across that double bond, we will oxidize it, and we will ultimately, okay, break this guy, 1, 2, 3. We're going to break this guy right here. When we do that, and there should be a couple of arrows here indicating we've got a couple more steps to get to here, we're left with this guy. Now, this guy's a little bit different. It's got 1, 2, 3, 4, 5 is where the double bond is, and it's an cis configuration. You might say, well, why doesn't the beta oxidation process just continue, and you could take away these hydrogens and electrons, but it turns out the enzyme won't touch this. This guy has got to be dealt with. Okay? And the way that this guy deals with this is, the way the cells deal with this is a little bit unusual. First of all, we see that there is the start of the process. The start of the process is that we do remove a hydrogen and associated electrons to make this, but this is a conjugated set of double bonds, and this is a different kind of a character than what we had over here. These are not conjugated, these are conjugated. Notice that this guy is in the trans configuration, this is in the cis configuration, okay? And another enzyme is necessary to deal with this unusual structure for the cell. This conjugated double bond is really a very different kind of electronic environment than we had over here. This is dealt with by the enzyme 2,4-dienoyl-CoA reductase. And what it's doing is two things. First of all, it's adding electrons from NADPH. So we know that we're going to have one double bond left over after we have finished this process. And the one double bond is not between either 4 and 5 or 2 and 3, but instead it's now between 3 and 4. So basically we've merged these two into one double bond, and that was possible because electrons and protons came from NADPH. And look what we did. We put them in the 3,4 trans configuration. Beautiful. Now we can use our enzyme, and we're proceeding along the way. With these two enzymes, we can oxidize every unsaturated fatty acid that we have in our body. Now, if you're using the 6th edition of the textbook, I will caution you that this structure that they show in the 6th edition is wrong. They show it in the cis configuration, and it's not. It's actually in the trans configuration, like you see here. Okay? Okay, so basically, now we've got something. Again, it's 2,3 trans. We can go ahead and actually, I said that wrong. I did say that wrong, didn't I? So this is, that's not in the trans. That should be in the cis, right? So it's still wrong. I just noticed that, okay? So in the 7th edition of the book, they didn't correct that error. This guy is in the cis because a 3,4 in the cis is what's needed for this enzyme. And they're showing it in the trans, okay? So actually, the 7th edition is wrong, as well as the 6th edition, all right? This actual structure is in the cis configuration, not the trans as it shows you. Okay, make a note of that. So that should be in a cis, not in a trans. The name is also wrong. Again, I'm not worried about the names, so don't worry about the names. Okay, well, we can now oxidize all of the unsaturated fatty acids that we have inside of our cells. Well, the last thing I need to deal with then is, or one of the last things I need to deal with on the oxidation side of things is to deal with what happens if I have an odd number of carbons. Okay? If I have an odd number of carbons, then there are other things I have to think about. So we can get down, let's say I started with a fatty acid that had 17 carbons. When I keep chopping, I'll get down to 5, and I knock off 2, I'll get to 3. And you might think, well, you can just cut that 3 into a 2 and a 1, but the enzymes won't touch that 3. So the 3, which is known as propionyl-CoA, has to be dealt with, and that's what I'm getting ready to show you right here. So how do cells deal with fatty acids that have 3 carbons? And the answer is shown right here. It's a very odd scheme. I can't emphasize this enough. This is a very, very odd thing that cells are doing not oxidizing, but in metabolizing propionyl-CoA. I hope to convince you of the oddness of this. Here's propionyl-CoA. it's got 3 carbons. And the cell says, oh, well, if it had 4 carbons, I can work with it. Okay, well, let's put another carbon on it. We can put a carbon on it very easily. And in fact, we can put a carbon dioxide on it very easily. So it'll be something like a fatty acid that we might metabolize. Okay, well, let's put this on here. So we grab a bicarbonate, we grab some ATP, and we put it on, except for instead of putting it on the end, we put it on carbon number 3. I'm sorry, carbon number 2. Okay? Well, okay, that's fine. So all we got to do now is move it. Well, no, not quite. Not only did we put it on carbon number 2, but we put it on carbon number 2 in the D configuration, and we really want it in the L configuration. Okay? Duh! What were you thinking, Sal? Right? Well, here's the L configuration, and then once it's in the L configuration, it's, oh, I know where I want this guy. It's on the end. Instead of going from here to here, it goes through here, here, and then over to here. This is an odd set of reactions, and it's an odd set of reactions that actually involves movement of a methyl group. This methyl group gets moved around in this process. And it takes an odd system to do that. Right? The odd system that does that, and by the way, once we get down to here, succinyl-CoA, of course, this now can be oxidized in the citric acid cycle. It can be used in the citric acid cycle. Okay? So at this point, that propionyl-CoA is now usable. That's what we're trying to get to over here, is something usable. Well, moving this methyl group around actually requires action of vitamin B12. Okay? I think you should have to draw this for the next exam, so please memorize this structure. If you get that cobalt in the wrong configuration, you're hosed, right? So don't forget the cobalt. All right, so you won't have to memorize it, of course. But B12 is an interesting coenzyme. Boop. I'm getting violent in my old age here. You thought I had too many beers before I came to class. I don't know. It still works. Okay. It's an unusual coenzyme. It's the only molecule that I'm aware of in our body that uses cobalt. Okay? And the cobalt actually plays a role in grabbing and carrying those methyl groups. Vitamin B12 is important for several things. This is one of the things that vitamin B12 is important for. Okay. You're not responsible for this mechanism. I'm just showing you what's happening in this process. So we're making some swaps. Okay. There's the grab of the what will become actually a methyl group. Right there is CH2. And there's the overall process. Okay. Well, so now we pretty much handled everything. There's only one thing I haven't told you about oxidation, and I told you about it, but I haven't shown you, and that is the fact that long fatty acids actually do get metabolized in a different location in the cell. Okay? So when I showed you everything coming into the mitochondrion, I lied just a little bit. Okay? You guys are used to professors lying. They do that all the time, right? Oh, this is going to be an easy exam, right? Okay. Well, the truth is that the long fatty acids start their oxidation process in an organelle known as the peroxisome. This is what a peroxisome looks like. It's normally not quite that large. And peroxisomal fatty acid oxidation requires that long-chain acyl-CoA dehydrogenase that I talked about. There it is. And this enzyme is only found in the peroxisome. So if you want to oxidize long-chain fatty acids, you have to first of all get them into the peroxisome, and when they go into the peroxisome, they get a CoA just like they got in the mitochondrion. Okay? Now, peroxisomal oxidation of fatty acids is not nearly as efficient from an energy perspective as mitochondrial oxidation of fatty acids is. The reason for that is because there's no electron transport chain in the peroxisome. There's no electron transport chain in the peroxisome, so the cell's got to have a way of regenerating FAD from FADH2. Okay? The way it does it is by action of this enzyme, of an enzyme that adds electrons to oxygen and makes hydrogen peroxide. Okay? Hydrogen peroxide breaks down to water and O2. Okay? But that means that the electrons that were here are not going in any reasonable fashion into the electron transport system. Okay? This guy can then go on through further oxidation, which it would continue to go through. And again, we're not going to have as much energy realized because we've got an NADH that we'll have to deal with as well. And there are shuttles it takes to actually get that out of the peroxisome into the mitochondrion, et cetera, et cetera. And that's a pretty involved process. So peroxisomal oxidation is not generating as many ATPs, all right, as mitochondrial oxidation is generating. Yes? Is that enzyme active whenever it's in the peroxisome? It's a good question. The answer is it is. Yes, it is. Okay, well, once the fatty acid gets broken down to a small enough size, let's say down into the 8 to 12 carbon length, it gets moved out of the peroxisome and it gets taken to the mitochondrion where the rest of the oxidation continues. Okay. Actually, it's even longer than that. So down to about 16 to 18. So once it gets down to about 16 to 18, it's taken to the mitochondria and is oxidized. Okay, so that's what fatty acid oxidation looks like. Before we move to the next topic, I thought we'd do a song. Okay? It's to a tune that you'll recognize because we used the tune, I think, the last time we sang. And it's When Johnny Comes Marching Home. The fatty acids carried by CoA are oxidized inside the mitochondria. They get to there, as you have seen, by hitching rights on carnitine. Then it goes away when acids get oxidized. Electrons move through membranes. Yes, it's true. It's true. They jump from complex 1 onto CoQ. The action can be quite intense when building proton gradients, and it's good for you when acids get oxidized. The protons pass through complex V, you see. They do this to make lots of ATP. The mechanism you should know goes through the stages LTO, so there's energy when acids get oxidized. Okay, that was a short one. Oop. All right. We turn our attention next to ketone bodies. Now, ketone bodies turn out to be relevant to fatty acid oxidation. And it may not be apparent at first, but I'll tell you why that's the case. So, your body, as you've seen, has a very large need for glucose. Okay? Glucose is our immediate source of energy. It's made by our liver, whether by breakdown of glycogen or by gluconeogenesis. What happens in those cases where glucose is not available? We do know that we go hypoglycemic sometimes, and when that happens, it's really nice and important to have a backup energy source available, because if we don't have that backup energy source available, our brain is toast, our eyes are toast, and probably we are toast as well. Okay? Well, the backup energy source turns out to be that bouncing ball. If we could just harvest the energy of that bouncing ball, life would be so much better. The backup energy source turns out to be ketone bodies. Now, ketone bodies are important because we can make them from fatty acids. So we're making a readily available, water-soluble set of compounds from fatty acids. That means they can be released quickly, and they can go and provide energy when needed, just like glucose can be made available quickly. Okay? Well, how do we make them? Okay? It turns out that we make ketone bodies starting with the last enzyme of fatty acid oxidation. Thiolase, you recall, split off a 2-carbon piece from a longer chain, right? Breaking between carbons 2 and 3. Well, what if that last piece has 4 carbons? It's going to make 2 2s, right? It's going to make 2 acetyl-CoAs from 1 4-carbon acyl-CoA, right? Well, all we're doing with this reaction is going backwards. So in the normal oxidation process, we'd be going right to left. In the synthesis of ketone body direction, we're going from left to right. Same enzyme, thiolase. So thiolase catalyzes that first reaction. It creates this molecule called acetyl-CoA. And that was actually what the last molecule looked like in fatty acid oxidation. A third set of 2 carbons is brought in with another CoA. And again, the CoA is split off, but the carbons are kept. And we create 3-hydroxy-3-methyl-glutarylcyl-CoA, or as we talked about it earlier, HMG-CoA. You saw this molecule when we were making cholesterol. HMG-CoA, as I pointed out at the time, is also an intermediate in the synthesis of ketone bodies. That 6-carbon intermediate is rearranged and a 2-carbon piece is removed, yielding this 4-carbon guy here, acetoacetate. That's one of the ketone bodies. Okay? It is a ketone, as you note. We see that acetoacetate has 2 possible fates, one that's useful and one that's not. The useful fate is shown going up because that's a reduction, and that reduction creates a molecule called a D3-hydroxybutyrate, or you can just call it hydroxybutyrate if you want. That hydroxybutyrate is stable. Chemically, it's stable. It's a nice way of moving this 4-carbon piece through our bloodstream. If acetoacetate doesn't get converted to that 4-carbon piece, it is chemically unstable. And it will spontaneously decarboxylate, okay, to yield acetone. It will spontaneously decarboxylate to yield acetone. Now, this turns out also to be of critical human health importance. Okay? The important piece of this is we're making ketone bodies when we're very low in glucose. People who have some forms of diabetes have real highs and real lows of glucose. And one of the ways in which they discover they're diabetic without any other indication is if you can smell acetone on their breath. If you smell acetone on one of your friend's breath, and I usually get a report of people who claim that they have that in this class like once or twice a year. If you smell acetone on your friend's breath, you want to tell them to go to the doctor and get checked to see if they have diabetes. Just because they have acetone doesn't mean they have diabetes, but they could. Okay? Because this means that their glucose levels have gotten very low, and the body is dumping ketone bodies out. Some of them are breaking apart in the lungs to produce acetone, which they exhale and which you can smell on their breath. All right. Well, I've talked about why this is an important energy source. I haven't told you how we get the energy. The brain needs energy. The brain needs glucose. The brain can't make glucose. And if you're starving or there's something else going on and you're not producing enough glucose, the body will start making these ketone bodies, which go into the bloodstream. Let's start up here. This guy's in the bloodstream. It makes it across the brain-blood barrier. I said that, didn't I? The brain-blood barrier. And then, what do? We reverse the whole process. We go from here back to here, from here back to here, here back to here. And finally, what we've done is we have delivered to the brain 2 acetyl-CoAs. What can acetyl-CoA be used for? Synthesis of ATP in the citric acid cycle. We just saved the brain. Okay? Clear as mud? Yes? Oh, your thumbs up. You approve of this. It's an election year, and I approve of this message, right? Okay. Okay, that's good. Let's see. What else can I say? That's a bunch of blah, blah, blah. And this is simply showing you the reversal of that whole process that I told you, except for I started with the... Well, hello! Please come down. Okay. Okay. let's move to fatty acid synthesis. Okay? So we've broken down fatty acids. We need to know how we make fatty acids. Okay? How do we make fatty acids? All right? Well, fatty acids, as I said earlier, chemically is very much like the reversal of the oxidation process. We're going to see there are some differences, but chemically, it's pretty much the reversal. One significant change, as we will see, and that's the very first steps in the process. Okay? Fatty acids grow by 2 carbons at a time during the synthesis process. However, this is a big however, the starting material is not a 2-carbon piece, it's a 3-carbon piece. So the cell is going to do something odd again in the synthesis of fatty acids. It's kind of like the odd thing you saw with the odd number of carbons. All right? This is going to involve an odd number of carbons in the synthesis process. Here's acetyl-CoA. Acetyl-CoA gets converted to a 3-carbon molecule known as malonyl-CoA, okay, by an enzyme whose name you absolutely need to know, and you're going to need to know something about how it works. It's known as acetyl-CoA carboxylase. Acetyl-CoA carboxylase. This guy, this enzyme, turns out to be the only enzyme in fatty acid synthesis that is regulated. It's the only enzyme in fatty acid synthesis that's regulated. Okay? All right. By the way, fatty acid synthesis is not occurring in the mitochondrion, again with a minor exception that I'll talk about later. It occurs in the cytoplasm. It's occurring in the cytoplasm. So oxidation and synthesis are physically separated in the cell. They're physically separated in the cell. All right. Well, in that process of making fatty acid, there's another difference from the oxidation, and that is the carrier. Okay? We saw the very first process occurs on a CoA, but that's the only place where we see CoAs involved. Everything else involves something called an acyl carrier protein. So in place of CoA during synthesis, we have something called acyl carrier protein. And though it may sound very different, if we look at what's attached to the acyl group, they're identical. Look at that. Okay? The thing hanging off of this protein is the same thing that's hanging off of coenzyme A. Okay? By having a carrier protein, the cell is able to recognize this is something that's being synthesized. Okay? It's a very visible difference because this thing hanging off here isn't present out here on coenzyme A. So this guy is being synthesized. Here are the synthesis reactions. Okay, so once we get it to this point, we have a malonyl ACP, and it turns out we have to have also an acetyl ACP because we have to have a starting material to start this fatty acid. We have to have a 2-carbon piece plus a 3-carbon piece to start. So this is priming the pump. This is getting everything set up. There we go. All right, so here's the 2 in blue. Here's our 3-carbon malonyl ACP. This is the other starting piece. What happens? First step is what we call condensation, and that simply involves attaching 2 carbons of that 3-carbon piece to the 2 carbons of the acetyl ACP. Follow the colors, okay? Here we go. The CO2 that comes off is this guy right here on the end. There's your CO2, okay? The 2 carbons of the acetyl ACP go on to the end. They go on to the end. So the attachment of this piece to this involves the loss of carbon dioxide. Okay, that's a condensation reaction that's occurring. Now we're home free because now that looks just like the end of fatty acid oxidation. The first thing we're going to do is we're going to reduce that ketone to an alcohol. We do that using NADPH. That makes a hydroxy group. Again, between carbons 2 and 3 is where all the action is. This one's in the D configuration. Previously we saw the fatty acid was in the L configuration when we were doing oxidation. All right, the next step then involves removal of water to make a double bond. That's a dehydration. There we go. There's our trans double bond. And the next step then involves hydrogenation, that is adding hydrogens and associated electrons from NADPH, and we're right there. Okay? Now there's a whole bunch of enzymes that do this, and they've got mouthful of names that are this long. All right? But the enzymes that do this are really interesting when we look at cells. They're really interesting. Why are they so interesting? Because they're all contained in one giant complex. One giant complex. And literally, the complex works like a clock. Okay? We see it at literally the fatty acid chain is moved around in a circular fashion with each of the reactions occurring as it goes around and around and around. Okay? We give one name to that big complex. It's known as fatty acid synthase, S-Y-N-T-H-A-S-E. So you don't have to know all the names of all the enzymes that do this because they're all contained in this one big enzyme known as fatty acid synthase. Acetyl-CoA carboxylase is not in fatty acid synthase. That's a separate enzyme. Keep that in mind. That's separate, okay? This guy here, okay, has all these other reactions in here that are important for the process to occur. Okay, so now we can make fatty acid. We've gone through one cycle. Now this becomes the starting material for the next cycle. So instead of having an acetyl ACP, we would have a butyryl ACP, and we would add another 3 to it. We'd add 2 of the 3. We'd have to have another malonyl to start. So malonyl ACP will always be needed on every round of the synthesis cycle. And on every round, one of those carbons is going to get lost. The newest material in the fatty acid as it's being made will be the material closest to the ACP. The newest material will be on this end of the molecule. The oldest material will be on this end of the molecule. Okay, let's see. What else did I want to say here? Reaction summary, blah, blah, blah. There's a whole bunch of different names. And you're going to call it fatty acid synthase. There's fatty acid synthase schematic. Oh, you don't want to see that either. But this, I just show you this. Okay, you don't need to know the mechanism. But this is the clock thing. You can see literally it's going around the hands of the clock. And this enzyme, this basic structure of this clock appears to be conserved across almost all of evolution. So the structure itself turns out to be very useful and very important in a variety of cells. Yes, sir?
So the fatty acid synthase within that enzyme not regulated, it's only that first carboxylase.
So the fatty acid synthase is not regulated. Once it starts, it's cranking it out. Now, one last thing I'll say, and then we'll call it a day. And the last thing I'll say is this fatty acid synthase works up to 16 carbons. The end product of fatty acid synthase is palmitic acid, or palmitoyl-CoA. Okay? We'll take it up next time from there.
Is that ACP always coordinated, or can that come out there and go back in and come out?
I'm not sure I understand the question.
That schematic showed ACP right in the center of the 4 pieces of fatty acid synthase. Is that always